protection against hepatitis B in Kalulushi District in Zambia. So the main aim of this project is to pilot a hepatitis B program to prevent hepatitis B infection in healthcare workers. So this is a 2-year project that began in December 2018 and is expected to end in December 2020. So under this project, we hope to raise awareness against hepatitis B, how hepatitis B can be treated as well as prevented. So we have enrolled a cohort of 673 healthcare workers. So these volunteered to enroll into the program, and we screened these healthcare workers for hepatitis B. And from our initial findings, you'll find that 66% have had a hepatitis B exposure. So basically that means in every group of 3 healthcare workers, at least 2 have been exposed to hepatitis B already. So what we did was we did screen for hepatitis B as well as measure our protective titers, that is the antibodies to the hepatitis B surface antigen. And so we found that only 24% had protective titers, but of these only about half had enough titers to last them even a year. So we then went ahead and offered the full vaccination course to those that were eligible and willing to get the vaccine. So basically that's where we are at, and we have followed this cohort. We are currently testing our samples in the lab so that we can know our post-vaccination findings.
One of the studies that is nested under the demonstration project on hepatitis B is a study that is trying to characterize T-cell responses in healthcare workers who've been vaccinated. So studies have shown that individuals who are vaccinated, that is adults, 90% of these adults respond to vaccination, whereas we have an approximately 10 to 15 who do not respond. So in this study, we're trying to understand response based on T cells and the role that they play. So T cells are actually the main cells that stimulate other cells in the body by producing cytokines, which then impact B cells to produce antibodies. So we actually study antibody titers as a correlate of protection instead of actually studying what's causing these responses. So as our first objective of the study, we are trying to study memory and effector cells and their impact on one's body to respond to the vaccine. And then one of our other objectives is to actually try to understand why the 10 to 15% of the vaccinees actually do not respond to vaccination. So we are going to do so by studying T-cell markers, which are senescence and activation markers. So once we know why this is happening, we'll be able to understand the immunological characteristics of our participants. Other studies have also shown that factors such as gender and age, including BMI, may affect how an individual responds to vaccination. So in order for us to understand our participants better, we'll be using these demographics and correlating them to how they've responded to the vaccine. And once we have obtained information on our vaccinees, we hope to influence drug development, and we also hope to influence prevention strategies and also response improvement in terms of vaccination. So we'll be able to determine whether our individuals need adjuvants, because we may need adjuvants to improve response in our vaccinees, because most of these vaccines have not been assessed in our population, which is Zambia. So once we get this information, we hope to influence policy and add to the body of knowledge.
I'd like to acknowledge our funders through the University of Oxford, as well as the Ministry of Health collaborators and other collaborators who've come on board to support this project. Thank you.